This e-poster is entitled Targeting the Stroma to Enhance Effector Memory T-Cell Infiltration and Anti-Tumor Response to Anti-PD1 Antibody in Pancreatic Ductal Adenocarcinoma. The authors have no relevant conflicts of interest to disclose. Pancreatic ductal adenocarcinoma, or PDAC, is a highly aggressive cancer that accounts for nearly 48,000 deaths annually and is resistant to many novel treatments, particularly immunotherapy, due to multiple major resistance mechanisms as highlighted here. It has been shown that focal adhesion kinase inhibitor can decrease immunosuppressive myeloid cells, consequently sensitizing PDAC to anti-PD1 antibody in murine models. Pancreatic cancer is a unique immunosuppressive and highly desmoplastic stroma with one of the highest HA concentrations among all tumor types. Our group has shown in a murine model that stromal targeting via pegylated recombinant human hyaluronidase, also known as PEG-PH20, enhanced the anti-tumor activity of whole cell vaccine GVAX by targeting CXCR4 expressing myeloid cells, as seen on the right, and led to enhanced survival, as seen on the left. Thus, in our current investigation, we evaluate the hypothesis that FAC expressing myeloid cells modulate T-cell infiltration in human PDAC, and FAC inhibition can synergize with PEG-PH20 by targeting myeloid cells in PDAC. Resected human PDAC treated with GVAX and anti-PD1 therapy was used to assess FAC expression in myeloid phenotypes and its impact on T-cell infiltration by employing multiplex IHC. An established murine model of metastatic PDAC treated with and without anti-PD1 antibody was used to assess the synergy and immune modulating effect of FACI and PEG-PH20. In human PDAC, by employing our multiplex IHC, we found that FAC is widely expressed in tumor-associated macrophages and neutrophils. Lower phosphorylated FAC neutrophil and lower phosphorylated FAC M2 tumor-associated macrophage density, but not lower phosphorylated FAC M1 TAM density, is associated with higher CD8 T-cell infiltration. By analyzing RNA sequence data from our GVAX and immunotherapy treated patients, we found increased FAC expression is associated with increased CXCR4 expression. By employing our metastatic murine PDAC model, we found that FACI and the combination of FACI and PD1 extends survival in mouse metastasis model of PDAC. Adding PEG-PH20 to FACI and anti-PD1 significantly prolonged survival in this model. Comparing the combination of FACI and anti-PD1, adding PEG-PH20 significantly decreased the number of CXCR4 expressing myeloid cells in PDAC. Specifically, we found decreases in granulocyte populations and tumor-associated macrophages. Consequently, this led to an increase in the amount of CD8 T cells and specifically effector memory T cell subsets. In conclusion, FAC is widely expressed in myeloid cell populations correlated with CXCR4 expression, and decreased FAC expression in myeloid inflamed stroma is associated with infiltration of effector CD8 T cells in human PDAC. Stromal degradation of hyaluronan via PEG-PH20 combined with FACI and anti-PD1 antibody depletes immunosuppressive cells in the TME, including GM-DSCs, granulocytes, and TAMs, and appears to target the CXCR pathway through PEG-PH20. These findings support the combination of FACI and anti-PD1 antibody with agents targeting CXCR4 directly or indirectly by PEG-PH20 in human PDAC. Thank you.